I want to start by extending my thanks to HM Payson. They have been sponsoring our Cafe Sci series for many years. They are a Maine-based financial advisor and have been just an enormous partner and supporter of ours, and we're very, very grateful for that. Okay, let's see. So for those of you that are watching virtually, as I go through the talk, you can use the Q&A button at the bottom of your screen. Type in your questions as I go along. I'm going to take a break midway through, and we'll do my best to answer the questions. There's also a raise hand and a chat button at the very bottom, but those features aren't working right now, so don't try doing them. Okay, so let's go back to the theme, creativity from chaos. So this past year has been a really wild ride on many fronts in the US. We have the pandemic, we had the racial reckoning that's been coming for a long time, we had the chaotic election. I usually travel extensively for my work, but like the rest of you, my world got very, very small, consisting primarily of my living room. Now at the start of the pandemic, as the CEO of Bigelow, the one thing I wasn't worried about was how to keep the science going. I worried about how to keep people healthy and whether to let people in the building, but I never really worried about the science itself. And that's because great science is like great art, and it can thrive during periods of disruption. Though it may take years to show up, all of the things that people are thinking about now in different ways in which they think. And just like a painter starts with a blank canvas, a researcher doing basic research is trying to uncover or discover or create new knowledge that just wasn't there before. And the circumstances that take us out of our comfort zone, make us think a little differently, those are the things that can really drive our creativity. Now, the pandemic caused us to look at a number of different approaches and to our operations, and we just looked at a lot of things differently, and we learned a number of very valuable lessons. We learned we could be very effective on Zoom. In this case, singing happy birthday to our board member, Barbara Burgess. I will do you all a giant favor and not play the audio of that. Our lab meetings became much more international. We started to invite colleagues from afar because why not? We gave more talks and invited people from far and wide to give seminars without the constraint of worrying about travel times and costs and logistics. So it really connected the science community in a way that we could have done before, but we didn't think about doing it before. The lack of travel and no commute really helped us lower our carbon footprint, which is a wonderful thing. Our dogs loved having us at home. That's Sunny and Tucker, my dogs. It saved us time in our commutes that we could put into making healthy meals and popping out for exercise. So it benefited us in a lot of different ways. Now, many of our staff will likely work from home going forward, much more often than they did in the past, but there wasn't all good. It was also much harder on some people than others. For people with young children, especially women, trying to work and oversee schoolwork was exhausting. I know how hard it was just to take care of 2 dogs that wanted attention. Having small children was just very, very difficult. And we saw some of our colleagues struggle and try to find ways to help them. As a nation, this is something we will be confronting for years in the future because it's estimated that as many as 4 million childcare slots have been permanently lost from the system that was already struggling to meet demand. So it wasn't all good. Another thing we learned is that successes are so much more fun than celebrating in person than remotely. And we have a tradition here. I'm looking down the hall at our bell. We have a bell on the Commons, and when we get really good news, we ring the bell, and we all gather around to ring it. And last Friday, we had our very first in-person impromptu bell ringing when I got a call from Beth Orcutt, who said, "Are you still in the building?" Beth has, there she is. We all gathered around the bell because she received a $2 million grant to start COBRA, the Coastal Ocean Biosphere Research Accelerator. Led by Beth, it will establish a virtual network of networks based at Bigelow, that will study the impact of deep sea industries, including deep ocean mining. So it's really important work, and it really, she has been and grown into such an incredible leader in the marine science community, and we are thrilled that she will continue to do this great work for the nation and the world. Am I pushing on this? I keep hearing a thump. Am I doing something, Stephen? No, okay. Then I'll move on. This past year was also time to really think about the next generation and how we prepare students. So Bigelow is collaborating with the Next Generation Science Exemplar Program. To date, over 10,000 teachers from 26 states have completed these professional development programs. And our senior research scientists, Kath Mitchell and Nick Record, and our Vice President of Education, Ben Twining, helped to develop a curriculum pathway on oceans, climate science, and big data. So really exciting. If you want to impact something, teach the teachers. So in the end, I am very, very proud how the lab had navigated the pandemic and how we really took care of each other. And thanks to the successes of our scientists at getting grants and contracts and the generosity of our donors, we ended the year ahead of budget, and we're even able to put some money towards hiring new scientists, which I will tell you about at the very end. So I think we got creative in the chaos, and as a result, we really had a great year. And I really look forward to seeing the fruits of this year of disruption as scientists and all the ideas they've had over the last year start developing these ideas moving forward. Okay, so now let's get to the good stuff. So at Bigelow, we study the foundation of global ocean health. And today I would like to do a deep dive into just what this means. So the foundation of all life in the ocean are the aquatic plants that known as algae. And that's where it all starts. And Bigelow is unique amongst marine labs in that we have this relatively narrow focus on algae. And it's one of the reasons we've been able to have the big impact we've had is because of that focus. Now, there are two broad types of algae. The first is microalgae. They're also referred to as phytoplankton. Most of these are not visible to the naked eye, and they range from tiny single-celled organisms to much larger colonial forms. They can have hard shells like these diatoms, or they can be soft cells like cyanobacteria. And we refer to these organisms as tiny giants because they are so critical to life on this planet. And for those of you who fly out of Portland Airport, you can see a whole series of electron micrographs and photographs of these tiny giants in a Bigelow display there as you're going down the escalator. And here are just some of the images that just blow me away. They're so beautiful. They're so complex. That's an electron micrograph of a diatom. These are mighty little animals. Actually, they're not animals. Scientists all over the world just groaned when I said that. So then there are macroalgae like brown, green, and red algae. These include the giant kelp, sea lettuce, rockweed that we see off the coast of Maine. As their name suggests, algae, both micro and macro, can come in a lot of different colors, and their color is determined by the pigments that they contain. And just like land plants come in a whole assortment of colors, all produced by a variety of different pigments, algae are the same. So here's a whole slew of different types of algae we have in our culture collection. And it's the diversity of pigments that's one of the reasons algae are of such interest for industrial applications. And it is this interest that we are trying to harness because in addition to studying the foundation of global ocean health, we also want to use our discoveries to improve the future for all life on our planet. It is this interest that of moving research into solutions that got me so excited about coming to Bigelow. And for years, I've been giving talks to try to educate people about climate change and the need to understand what's happening, but that's just not enough. We need to take what we learn and get products and approaches into the market at scale if we're really going to make an impact, not only in understanding climate change, but solving the problems that it's creating. So today I want to tell you a number of those ways that we do that. And it all starts with algae. And got algae? Yes, we do. So Bigelow is one of the top labs in the world for the study and manipulation of algae, starting back from its very earliest days. When I was a graduate student, I bought algae from Bigelow. So it has a long tradition. Not that long, I'm not that old. So to start, let's remind ourselves just some basic biology. So I apologize if this is old school for many of you. So like all plants, aquatic plants take up carbon dioxide and they take up nutrients and they grow and produce more of themselves. And this is true for both micro and macroalgae. And using the raw materials of CO2 and nutrients and energy from the sun, these tens of thousands of different types of micro and macroalgae can produce just an enormous array of compounds within their cells. Now, Bigelow is home to the National Center for Marine Algae and Microbiota, which receives and maintains and distributes living cultures of marine freshwater algae, bacteria, and viruses. Back in 1992, the Oceans Act designated NCMA the National Marine Algal Facility. Incidentally, just because it's a national facility doesn't mean the nation pays for it, unfortunately. We have to figure out how to do that ourselves each year. But within this collection, we have cells that can do just an amazing array of biochemical processes. And then we have Bigelow scientists like Jose Robledo, who's a synthetic biologist, who can use genetic engineering to insert genetic information that a cell needs to produce just almost an unlimited array of compounds. And in this sense, I would love to get you to start thinking about algae as chemical factories. And they are factories with a very, very important attribute. Their raw materials, carbon dioxide and nutrients, are some of humanity's biggest waste problems. So in 2019, the US produced roughly 5.2 billion tons of carbon dioxide. That's not greenhouse gases, that's just CO2 alone. And about 18 trillion gallons of wastewater. We are literally drowning in the raw materials that algae need to grow. So in the past, humanity has had a very linear economy where we take natural resources, we make something, usually in a giant factory that's located in one area and gets bigger and bigger and bigger, uses fossil fuels to do it. And then we dispose of whatever we produced, either by tossing it in a landfill or unfortunately pouring it into the ocean. And what humanity desperately needs to start thinking about is holistically how we can deal with manufacturing from the start of the process all the way to the end. So in the future, we need to develop much more of a circular economy where every step of the way we are thinking about the next step and how to make it sustainable, and then the step after that and after that. And consistent with the theme of this talk, while we're emerging from this whole year of chaos, this is a great time to get creative on how to rebuild in the future. And algae can be an important part of this transformation if we start thinking of them as the factories of the future. So how would this work? And I'm going to just walk you through a very simple example. So here we have our circular economy. Let's start with one, the factory. Instead of chemicals being produced in a giant traditional localized factory powered by fossil fuels, we would have algal farms that are solar powered, both from the perspective of their operations, right? Because it takes energy to set up and power these kind of things, but also the energy source for the algae. They would be smaller, they would be much more distributed around, and perhaps much more focused on a smaller number of products. So let's go to the second phase. So what do they produce? They could produce any number of things. Two things that I'm going to focus on in my talk today are supplements and dyes. So let's go with something like spirulina, because actually it is algae, so it's probably not the best example, but I'll give you many more in a bit. So it produces this, and then what's left over? Let's go to the third point, and that's the recycling part. At the end of the process, you have what? You have dead algae, which is an incredible fertilizer. So instead of mining phosphorus and all the energy and environmental degradation that causes, and creating nitrogen fertilizer in giant factories with the Haber-Bosch process, we are producing great fertilizer in our algal factories. To start the process over, let's go back to that wastewater treatment plant and the excess nitrogen and phosphorus it was not able to remove. In so many parts of the world, we do a very poor job of treating wastewater, but that instead of treating it as a waste, let's treat it as a resource, and it could feed right back into our algal factories. So in a circular economy, we don't think about waste we need to throw away. We think about resources we need to use to our benefit. And now I want to go back and visit the products that you could make by doing a deeper dive into supplements and to revisit this idea of distributed factories. So the global supplement market was worth $168 billion in 2019. These are the supplements like the hair and nail vitamins that I take to make my hair and nails long, despite the fact that I keep my hair up all the time and keep my nails short, right? So this is at times an irrational supplement market, but it is a huge supplement market. Before the pandemic, it was estimated to reach a value of $307 billion by 2026. There is a massive desire for supplements of all sorts. So here's the example I used before, spirulina. It's a blue-green algae that is a common supplement in smoothies. If you've bought any kind of green drink at anywhere or in the supermarket, it probably has spirulina in it. Unfortunately, with this, as with any supplement on the market today, you don't really know what you're getting because the Food and Drug Administration doesn't verify supplements, what they contain, or what, especially the things you might not want them to contain, like heavy metals, bacteria, pesticides. But what if you could grow your own? So the picture on your right is a picture of a prototype that Mike Lomas and his team created for a self-contained algal factory that you could have in your home. This could be scaled down. It's automated. All you need to do is have the media, the growth media, which is basically the fluid that the algae are growing in. And the system does the rest in terms of harvesting your algae that you could use in your shakes. They're refining this to make it automatic. And if you think this sounds wacky, think of all the people that are growing vegetables every day in their yard. This is just another food that you could be growing in your home. And in the future, people could be growing their own vitamins, their supplements in their window, and there's all kinds of great business opportunities that could enable them to do that. And best of all, you know exactly what's in that product that you're producing yourself. So this is just one commercial use, and I'll go into a few others. But before I do that, I am going to stop and pause and ask if there are any questions. And remember to use your question and answer button at the bottom of your screen to submit them. Yeah.
Oh, can you grow the algae in the ocean, or is it only a land-based growing process?
So the problem of growing them in the ocean is how would you keep them all together? You would need, I guess you could put a tank for temperature control, but you would want to have a solid tank because they're microscopic. They're not like fish that you can put in a net, right?
Okay.
Chris.
Is Mike Lomas's greenhouse producing algae for sale, or is it being used at the lab?
I'm actually going to go into that. Both. It's actually both. So we have contracts that we produce lots of algae for businesses. A lot of what we have, we are doing for ourselves for our own research. Louise?
What would make the biggest difference for the circular economy? Oh, what would make the biggest difference for the circular economy would be algae that could become plastic.
Yes. Here, here, here. Okay. I see. Okay, so I am, okay.
I'm asking questions on behalf of the online audience. Richard Bentz asked, is the teacher resource available to see or use?
Is the teacher resource available to see or use? I'm not sure. Actually, can you send me an email and I will pass that on to Ben Twining, who's been very intimately involved. I don't know how proprietary that is. I hope it's not. And the answer is yes, but I don't know that for sure. So send me an email.
All right. And then the last one is, I've heard of seaweed and kelp having a positive impact because it is carbon negative when you drop it back into the ocean. Is this something that could be achieved using algae as well?
So I think it's a bit of an open question on the kelp. I'm not sure. There's some debate in the science community whether it is carbon negative. A lot depends on the idea of trying to make kelp carbon negative is you raise it in platforms out in the open ocean, and then you sink it down to the bottom. And because while it's growing at the surface, it's sucking carbon dioxide out of the atmosphere that has been dissolved in the water, and then it takes that and is released. A lot of it depends on how long it stays down there, what happens to it. And I don't think there's robust data right now to show that it is taken out of the system long term. So I will just have one cautionary thing about that, whether that is actually a net carbon sink, if we can say that, and if we know the magnitude of that. For phytoplankton, there's a lot of modeling work that have been trying to show what the fate of that phytoplankton is. One of the things we do know is that the shells, like in the pictures I did early on, is the shells that have the phytoplankton cells that have shells, like the diatoms, they're heavier. They are much more likely to sink deep into the water column. They also are bigger, tend to be bigger, and so they can be eaten by organisms that then, when they make fecal pellets, the scientific phrase for poop, it has a much better chance of sinking down into the deep ocean. So it really depends on the kind of phytoplankton, whether that is going to be transported into the deep ocean. Get my camera. Any more? Okay, so let's keep going. Okay, so commercial uses. What are some other commercial uses of both micro and macroalgae? So one great one is organic fertilizer. You can use macroalgae, and it has been used historically as fertilizer, stabilizers and emulsifiers. So if you've had ice cream recently, many ice cream brands use macroalgae as the emulsifier. Biogas, feedstock for biogas, or both micro and macroalgae have been used for biofuel. You can just eat them, right? Spirulina, you can eat it. Macroalgae, there's a whole number of, especially in the state of Maine, there's companies that are producing delicious kimchi made out of macroalgae. So you can use it in many different ways. And Americans are kind of unusual in that we don't eat more of these macroalgae. If you go to Asia, they're in the diet all the time. It can be used as an animal feed. Bigelow has a large $12 million project that is working to develop a mixture of microalgae and macroalgae as animal feed to reduce enteric methane. So that's the production of methane by cows. Bioplastics, which is what we just heard in the question and answer. So if we could produce at scale for the market bioplastics made out of phytoplankton, what a boom that would be for humanity. Microalgae is also used in nutraceuticals, pharmaceuticals, cosmetics, minerals, vitamins can be obtained from this, and colorants. And colorants is one of those commercial uses that we will, I'll be talking about in a minute. So we believe so strongly in the potential of algae that in our current strategic plan, Bigelow established the Center for Algal Innovation. Now, this is headed up by Mike Lomas, who also runs our National Center for Marine Algae and Microbiota. It has a long mission statement, but the key parts of it is we want to connect researchers with entrepreneurs so that we can create a sustainable, ecologically sound, and profitable algal value pyramid. Starting with this in Maine, but really this is something that we should be connecting people across the US, through New England, and around the world. This is something that all of humanity could get involved with. So the center does a lot of different work. It does contract research to answer the question that we just had. It does biomass production. So these are some tanks where we're growing large volumes of algae. We do this sometimes for our own experiments, but in this case, we're hired to do it by a company. Companies can hire us to screen compounds. If you want to use algae to make certain chemicals, with our analytical facilities, we can help you do that. And also to optimize the media. So the media is what the algae are growing in, it's the fluid. And depending on what's in that fluid, it can produce different compounds, grow at different rates, and can really add to the array of different things you can do with algae if you know how to grow them different ways. We also have incubator space. This is from bench scale labs in our main laboratory building to pilot scale greenhouse. We have an application lab, a fabrication lab, where we can work with entrepreneurs to help them with their own companies, and we also can provide some tech transfer support. So we're really trying to engage people in this space. And because we're interested in building and supporting the algal economy, we want to train the workforce that's needed. So Bigelow has a very long history of doing this for the marine science field. When I was a researcher in Virginia, I sent my lab manager and one of my technicians to Bigelow to be trained. They took, I think, a total of 3 classes. So we could do this with our existing courses, but also additional professional training programs and new trade courses. And then we do our own internal research through this. So one is, developing algal growth systems like the one that I talked about earlier, working on various ways to address problems in sustainable aquaculture in partnership. Many of those are in partnership with the Center for Seafood Solutions, and also in bioremediation. So one area that I mentioned is that the Center for Algal Innovations is working on is colorants. So colorants are used in food and cosmetics, as well as a number of other industries. And if you look at the back of most packaging, you will find an array of different colors. So yellow number 6, red number 40, yellow number 5. So these colorants come from a variety of different sources, some natural, some industrial. If you're my age, you will remember all the hoopla when I was a kid of the red dyes causing cancer. So depending on where you get your colorants, they're not always the healthiest thing to be consumed. But I recall at the start that I said that the different types of micro and macroalgae naturally produce a wide variety of colored pigments in their cells. And here are some of the examples of the pigments produced at Bigelow. And they all start with algal cultures. And these pigments can be extracted and used to color food products or anything you would want to color or dye things with. In many cases, the type of pigments an algae produces is dependent on the conditions they grow in. So for example, you might recall this image from a Bigelow holiday card a few years ago. This is a culture of Hematococcus. And Hematococcus, when it's healthy and happy, it's green. And when it starts becoming stressed, it produces a lot of a red pigment called astaxanthin. So if you know how to make your cells unhappy, you can produce a whole lot of astaxanthin in these cultures. And you all know astaxanthin, though you might not realize it, but it's if you eat something with astaxanthin, like a flamingo, turns it pink. It's the reason why salmon meat is pink. So it's a very common pigment that shows up in food webs. So Mike and his team are working to produce a number of pigments using other species like Rhodomonas, which is this guy, and Proteomonas, which is this guy. The important thing is that these dyes are not produced with fossil fuels in a factory. They're produced by algae using sunlight. In a circular algal economy, you could grow algae for some other reason and harvest the pigments just as an added bonus. And here again, I want to bring back this idea of creativity. When you think about algae, the algae industry is not going to be a straight line where you build an algae farm to produce product A. The economics will likely require that you get very creative and think holistically to maximize all the potential products that come out, as well as treating what's left over at the end as a valuable resource. So I want to give you one final potential use of algae, and it involves micro and nanoplastics. So when people think of plastic in the ocean, we usually picture beaches strewn with plastic bottles. It's terrible. But in reality, this is not where most of the plastic in the ocean is and what it looks like. Upwards of 99% of the plastic in the ocean is in the form of micro and nanoplastics that you will likely never even notice. They are everywhere in the air around us and in the deepest parts of the ocean. In a recent study that came out, I think it was just this past year, I feel like I lost a year in the literature. I think it was in the past year. It was estimated that Americans consume 74,000 pieces of microplastic every year. Another study framed it as the equivalent of a plastic in a credit card. So it's all around us and it's in us. Plastics come from a lot of different sources. So the biggest source is synthetic textiles, car tires, city dust, personal care products, road markings. The synthetic textiles, I have a polar fleece story, and then in the Arctic. occasionally, when I was on this big project in the Arctic, we were up there for I think 5 weeks, and we would filter at the end of our experiments. And every so often, we would have these long grain strands that I'm outing myself as a chemist here because I was like, is that a colonial phytoplankton? And then we realized, and I would dutifully note it down in my notebook when we would see these and trying to figure out what kind of conditions are these colonial phytoplankton showing up, till I realized they only showed up when I wore my green fleece. So these things really are just everywhere. I thought I'd get a little laugh from that from the local, but they don't have to if they don't want to laugh. But I got a pity laugh there. Actually, it wasn't funny. It was very alarming because we had to go back and remember what samples we had those in. So working with summer interns, 2 of our senior research scientists, Patty Matrai and David Fields, published a study with microplastic fibers and showed how they can accumulate in the guts of larval lobsters. So organisms, just like we see pictures of seabirds that have eaten large pieces of plastic, the organisms in the ocean are eating these microplastics, which is a problem because they get no nutritional value from them. In another previous study, Patty and her team showed that mussels also accumulate these microfibers, and that's a problem because we end up eating those microfibers when we eat the mussels. They also showed, though, that if you put mussels into filtered seawater, over time, they will clear themselves of much of those fibers by packaging pseudofeces, which again is just basically pseudo poop, using a bunch of mucus. But the problem there is that if they're making pseudofeces, they are not putting that energy into growing or reproduction, which if you're a mussel farmer is what you want them to be doing. So the bigger problem is how do we get them out of the environment? So recall I said earlier, this is our wastewater treatment plant. The US produces 18 trillion gallons of wastewater every year. And during the treatment process, many of the microplastics get removed in the sludge. And the rest leave the plant and go back into the environment. And those that aren't removed, many of those are filaments. Those are the ones that were appearing on my filter. So Mike and his team are working on a way to remove these. Let's go back to our algal cell. While the cell grows, they can produce a lot of different things. And one of the things they produce is sugars, also called mucopolysaccharides. And these are released in the water around them. So if you've ever been at the beach and you see that kind of frothy sea foam, that is mucopolysaccharides. That's sticky compounds that are making these bubbles. The thing about sugars is that they are sticky, and that's the characteristic we want to exploit. So here's a video of some material that's produced by the Center for Algal Innovation. And this is harvested microalgae that, because of all the sugars it produces, looks and behaves a whole lot like Jell-O. Now, there could be a lot of uses for this stuff, and the one we're talking about now is as a removal mechanism for microplastics. So a project that the center recently started with a summer intern, Andre Martin, who is with us from Haverford College in Pennsylvania, is looking at how algae and the sugars they produce could be used as a water polisher to remove these microplastics in domestic wastewater before it's released into the environment. So the algae would be grown along this raceway, which is basically just a trough. And then the water is passed over it. And as it passes over that algae, the microplastics stick to it. So here's a shot showing that sugar material with microplastics that have been collected on it. If you shine UV light, these microplastics show up and fluoresce. So all those purple dots are the microplastics that have been stuck onto this material. So the idea is that you have a wastewater treatment plant, that the material comes in, goes through these various treatments, and here's the water. This band is representing the water. It has these microfibers in it, but it's clearing up over time as it's going through treatment. And then in the final stage, it would pass over this algae. Isn't that cool? Mike Lomas made that, and then I messed it up, and it took me like 45 minutes to figure out how to fix it again. So I hope we're going to do that one more time. There it is. What is really exciting to me about this, and this is still very early stage research, but what is really exciting to me is microplastics are one of those things that I despair of at 3 o'clock in the morning when I can't sleep. And this is one of the first things that I really thought you could do something with that. because the problem is so intractable, and yet with some creativity, maybe it's not. So to wrap things up, I want to leave you with 3 images. First, the idea of algae as factories of the future. You know, algae are mighty. Without them, this planet would be a cold, dead rock, and none of us would be here. So if they can do this to Earth, think of what they could do if we really put our mind to harnessing all their power. The second is this idea of a circular economy, where there's no start and no end, and where each step along the way, you need to consider where you're starting, what resources we're using, and what you're producing. And everything has to be considered to have value. And to do this, we're going to have to get creative. And what better time to get creative than at the end? Well, it's not the end of the pandemic. We're starting to see the beginning of the end of the pandemic as we emerge from this big global disruption. And finally, I want to leave with you that great science is like great art. I believe both are beautiful, and by their very existence, they make the world a better place. So before I introduce the rest of the cafe side talks in a minute, I'd be happy to take any more questions.
We have some from online.
Okay.
The first is, has your staff grown in the past couple of years, and has it added more female and minority scientists?
So our staff has grown. I don't know by our net. So one of the things is some of the scientists that we have are, like, we have a lot of postdoctoral scholars. They come from anywhere for 1 to 4 years, say. And so during the pandemic, that was some of the turnover. I think we hired 22 people last year. And a lot of those were postdocs and staff. So yes, we have hired, and I was very proud that we continued to hire during the pandemic. So we were a bright spot in that kind of dismal landscape for a long time. In terms of hiring female scientists, so one of the things we did this past year is we had a big search for new senior research scientists. And I'm going to call out Beth Orcutt again. So Beth is our diversity, equity, and inclusion liaison. And she worked with our chief of staff to really take a look at our hiring practices. And one of the things we did was to create a rubric where we had to evaluate all of the candidates. We had 113, I mean, virtually all of them were really good candidates for a senior research scientist position. So you could no longer just do a gut, oh, I think they're really good, they're excellent, they're good, they're very bad. You had to specify a reason for each of the rankings of each of the various ways that we rated these candidates. And we were trying to remove unconscious bias. So in the end, we did not set out to hire a lot of women, but in the end, as you will see in a minute, most of the people we hired or promoted were actually women. And which I was thrilled about. But I'm even more thrilled that we didn't set that up as a goal. We wanted to hire the very best scientists, but those ended up being women. Doesn't always happen, but it happened in this case. So yes, we have expanded. Yeah. Any more questions?
Yeah, we have more from online, but if anyone has questions in the room, just raise your hand.
Good evening. Is Bigelow doing anything in conjunction with the state of Florida on the blue-green algae problems coming out of Lake Okeechobee and causing trouble in the estuaries on both sides of the state? We also are experiencing a lot of red tide issues from phosphates and other discharge out of Lake Okeechobee.
Right. So my own research, I'm a nutrient biogeochemist. I study nitrogen, which is a big part of the red tide issue. And I spent 15 years studying the red tide. So we can talk after this. We also have Pete Countway, the senior research scientist here, has a project down with Cindy Heil, who is heading up their Caridia brevis task force down there. She used to be at Bigelow. So we are doing work down there, and we just hired, and one of the people giving the last cafe sci is Rachel Sipler. We are starting a water health and humans initiative. And one of the things to look at are harmful algal blooms, because the species that are causing the problems in Florida and on the West Coast are also starting to appear in the Gulf of Maine. So the answer is yes. Yeah, that's my attitude. He just made a shocked face. So one of my own personal goals, and one of the reasons I'm thrilled with Water Health in Humans as a new initiative in our strat plan, is in a place like Maine, I think we're catching it early, and we have the chance to do it right and not have it turn into the Gulf of Mexico. But the answer is yes, we do have people working on that.
All right, another one from online. Once the algae collects the microfibers, then what do you do with the resulting algae plus microfibers? How do you dispose of it or get rid of it?
So that was a question that I got yesterday when I gave a practice talk to the staff. I don't have a good answer for that, other than because I did not follow up with Mike, who's the guy who's heading this up. The simplest thing would be to filter out the microfibers, and then you've got just the algae that you could use as fertilizer, whatever. Or the simplest thing is you just bury the algae in the microfibers. But at least it's not going into the ocean. But yeah, you can't, at this point, there isn't a way to degrade that plastic. But I don't know if any of you read Science Magazine, which is one of the kind of two science journals that talks about all, the state of the art across all disciplines. And every few months, there's another article of encouraging results about ways of, one, genetically engineering microbes to degrade plastic, to develop plastic that naturally breaks down under certain conditions. And so at this point, I would take just being able to capture it and get it out of the ocean would be a huge bonus. But what exactly you could do, I'm not sure.
You've given us a lot of interesting examples, kind of inspiring examples of solutions, potential solutions to a number of issues, but they're all on a small scale. What would be involved in scaling some of this up? Because most of these are global problems, which require huge scaling. How do our scientists work with companies, or do they do it themselves? How is it going to scale up so that it actually has a meaningful impact on the globe?
So I think one of the things we need to start doing is to kind of rethink the scale of what makes a difference, right? So think about our electric grid. So we have grown our in the United States by building these massive power plants that then have power lines that are sent, out in all directions. In reality, that is an incredibly inefficient way to do it. And that's because when you are moving electrons back and forth, you will lose some of those electrons and the energy that's involved. So we would have been much better off if we had a distributed series of plants, power plants. And that's how we should rebuild with solar, right? So we rebuild solar plants. It's not going to be one massive solar plant. It's going to be lots of solar plants. I think the same thing is true with manufacturing. We had manufacturing centers in our country where we would build massive factories. What did that require? That required you to bring raw materials from very far away. And so distributing it. So just thinking about the scale of what is the kind of industry we could have just by making it more distributed and smaller in scale, there will be certain efficiencies surrounded with that. But that also means, and that's where the creativity will come into. So for example, I did some work way back when I was involved in a project on biofuel. We were going to try to raise algae that had a very high lipid content so we could create biofuel. And it was actually a colossal failure, actually. And part of the issue was it was not going to be cost effective. And what we really needed to have done is looked at all the other things we could have gotten out of that material that we were producing and not just focus so much on the lipid. So for example, there are certain compounds are very expensive to produce. And their resin technology is really good. You can pull out a lot of different things that you want out of algae. And by genetically engineering algae to make the things you want, the lipid is just one one part of the puzzle because we're producing all of these other streams, but it's a much more complicated way of looking at things. Like I alluded to the Burp Buster project where we're trying to reduce enteric methane. At the end, how this project is evolving is there's going to be so many benefits to the cow of what's being produced that the farmers aren't going to do it to reduce enteric methane. That's the added bonus that we were really interested in. So again, we've got to think much broader on all of these and not just in this linear, we're going to do this to get this. We're going to do this and it's going to produce this, and we got to figure out what to do with all of it.
We've got a more technical question. Are you aware of any experimentation using algae to remove PFAS compounds from the water?
No, I am not. Are they?
Another technical one. Can algae derived, and I'm going to butcher this pronunciation, astaxanthin be used for research requiring fluorescence, alternative to chemicals such as red, Nile red, etc.?
Could astaxanthin be used for fluorescence?
For research requiring fluorescence as an alternative to chemicals such as Nile red.
I don't know if astaxanthin fluoresces. I mean, the fluorescent particles we were showing, that was plastic. So I'm not sure. I don't know what wavelengths astaxanthin might fluoresce at. So this is why I would tell my students, you're going to get questions you can't answer, and I just got 2 in a row. So, no, I don't know.
Last call for questions in the room.
Probably somebody I went to grad school with that's going to prank me by calling into all of these and asking me questions. Okay, so then I'm going to go on and just very quickly, I want to tell you about our upcoming talks where we will highlight our new or newly promoted scientists. First up next Tuesday is Kath Mitchell. Kath is a physicist from Scotland who was recently promoted to senior research scientist. So she originally came as a postdoc, then became a research scientist, and is now a senior research scientist. Kath is carrying on the tradition of cutting-edge research in satellite oceanography with her talk, Beyond Blue, a study of ocean color from above and below. Next, we will hear from Maya Groner. Maya will be joining Bigelow in November. She is coming from a research institute in Seattle. She will be joining us remotely, because we're used to Zoom, to describe her work in lobsters in a pinch, impact of warming oceans on epizootic shell disease. On August 3rd, we will hear from Nicole Polton, who was recently promoted to senior research scientist, long overdue, and we are so thrilled. She is the director of our Center for Aquatic Cytometry and is doing such important work to provide new tools to study microplastics, because right now we are really hampered in research on microplastics because they are so difficult to quantify and deal with. And she will tell us about her progress so far in persistent plastics, using familiar tools and new ways to explore the impact of microplastics. Then, in a great example of how we have learned to take advantage of the virtual world, on August 10th, Alex Michard will be providing his cafe side talk from Tulik Research Station in Alaska. Alex was promoted from a postdoctoral scholar to a research scientist and will be telling us about the life of ice, the global influence of microbes in polar regions. And then rounding out the series will be new senior research scientist Rachel Sipler. Rachel just joined us a few weeks ago from Memorial University in St. John's, Newfoundland. She is the director of the Water, Health, and Humans Initiative that I mentioned earlier in our strategic plan, and she will be sharing her initial thoughts in her talk, Water, Health, and Humans, a new initiative to protect Maine's most valuable resources, which are people and water. So with that, I want to thank you all for joining us tonight and for caring enough to log on and to learn about the ocean and what Bigelow is trying to do to protect it. I hope to see you all next week for our virtual presentation, and have a very, very good evening. Thank you.